So very good afternoon to all of you, all the participants. I am very happy to be the part of this PhD coursework program. At the outset, I would like to thank the organizers for giving me this wonderful opportunity to interact with the students. So I will be giving two lectures. The first part of my lecture will be confined to UV visible spectroscopy, and the second part of my lecture will be fluorescence spectrophotometer. Okay. So before exactly the principal instrumentation and the application part of UV visible spectrophotometer, I try to give you some fundamental information which will definitely help us to understand better what exactly this spectroscopy is all about. If you look at the basic source of energy, okay, as we are all quite familiar, sun is the principal source of energy. So when sun emits light of a different wavelength, okay, some of the wavelength, the light emitted by the sun, they are reflected as short wavelength radiation. Some of them are emitted as a long wavelength radiation also, of which some of the wavelength of light can be absorbed by the Earth's surface, preferably by the plant system. So the principal source of energy is sun. The light is considered to be a electromagnetic radiation because when you take a normal white light, it consists of two components. One is electric component, another one is the magnetic component. In the case of normal light, both electric and magnetic component, they are travel perpendicular to each other. This is the point number one. The point number two is, since it is made up of both electric and magnetic component, the whatever wave light is light which is emitted by the sun is considered to be electromagnetic radiation. So light travels in the form of waves as well as particle. So light is being represented in the form of wavelength. So as you can see from this picture, it is very easy to understand what exactly the wavelength is. So the wavelength is basically a spatial distance between two different peaks or two consecutive peaks in the sinusoidal waveform. This is considered to be a sinusoidal wave. I hope you are all familiar with the sinusoidal wave. So the distance between two consecutive peaks of the sinusoidal wave is considered as a wavelength. So sun emit light of different wavelengths. That's what the point I would like to emphasize here. Okay. So when light of particular wavelength interact with matter, so what is happening actually? So the interaction of light with the matter, if you take any biological material, that biological molecule is considered as a matter as of now. Okay. Right. So when the light interact with this biological molecule, when the light is interact with this matter, it is considered to be a quantum phenomena.
Sir,
Yeah.
can I disturb you for a second?
Yeah.
Actually, what is happening? Your voice is actually breaking.
Breaking. Okay.
Probably it is due to the headphone. Actually,
Headphone.
probably it is due to that because when we are speaking,
Okay.
the
Is it clear
headphone may
now?
be. Yes, it is clear now.
Okay. If you have any
Okay, no
problem with
issue.
my voice, kindly stop me. Okay.
Okay, okay, yes, yes, I will tell you.
Okay.
No issue.
So when the light of definite wavelength interact with any matter, let us take any biological molecule, the light possesses some energy. This energy is being absorbed by the particular molecule, biomolecule of interest. The valence electron is getting absorbed some light energy and it is getting moved to a higher energy level. So during this process, what happened? The light energy is being absorbed by a molecule, biomolecule. That biomolecule, the valence electron of this biomolecule tend to move to the higher energy state. Initially, it is in the ground state before absorbing light of definite energy. Once it absorbs light of definite energy, these valence electrons in particular move to a higher energy level. So let us make it very simple. When the light impact on a particular biomolecule, the biomolecule is capable of light of definite energy. So not all biomolecules are capable of absorbing light of definite wavelength. For example, if you take a Bradford reagent, okay, it will absorb light after interacting with protein. It will absorb light of only 650 nanometer wavelength of light. Okay, so each biomolecule has a peculiar property of absorbing light of definite wavelength. So this is what happening when the light interact with any biomolecule. So each biomolecule is capable of absorbing light of definite wavelength. Not all biomolecules are capable of absorbing light of all wavelengths. So it is very, very specific. So the interaction between a light and biomolecule is very, very specific. So when a light hits on an object, let us take the biomolecule, there are three possibilities. The first possibility is the light, incoming light in the form of wave, it can be reflected or scattered, or some amount of light can be absorbed by the biomolecule of interest. The third possibility is that whatever light that is impinges on this biomolecule, they can be transmitted. So once you know the quantum of light that is being transmitted or impinges on the sample, depending on the depending on the quality of transmitted light or quantum of transmitted light, you can easily quantify how much light is being absorbed by the biomolecule of interest. So when light impinges on the or when light hits on the particular object, there are only three possibilities. It is either absorbed by the biomolecule, or the light can be transmitted by the biomolecule of interest without any absorbance. The third possibility is the light can be reflected. Okay, in that case, there is neither absorption nor transmission. So, in case what happens, if no light is being absorbed by a biomolecule of interest, it appears as white. That means all light, whatever is trying to impinges on the biomolecule, all the lights are being reflected back. Nothing is absorbed. When all visible light is being absorbed by an object of your choice, then the material appears black in color. Some of the shirts look very dark in color, especially black in color, which means what? All light that is being absorbed by this shirt of your choice. Okay. So depending on the quantum of light, depending on the quality of light that is being absorbed or either reflected or being transmitted, the color will appear. Okay. So the apple is a very popular example, right? An apple appears red in color it because depending it leaf reflect only mostly green light to the eye, but apple reflects mostly red light to our human eye. That's why we could able to see the red color of the nice red color of the very attractive red color of the apple, whereas leaves are looking very green because leaves reflect mostly green light to our human eye. Okay, so everything depends on the interaction of light with the biomolecule of interest, right? So there are two basic theories actually which supports the nature of light. Scientists keep asking question, what exactly the nature of light? So there are two theories which are very popularly known to the scientific community. The one is called wave model. The second one is first one is called wave model. The second one is called particle nature of light or particle model. So the wave model of was supported by a group of scientists, very popular physicists, I would say, Maxwell and Hertz, whereas the particle model was supported by Albert Einstein and also Max Planck. Okay, so now we know for sure that light has a dual property. Light has a dual nature. Light behave in the form of wave. Light behave also in the form of particle. So there are particle nature of light and the wave nature of light. Both are accepted now. So if you look at the wave model of electromagnetic radiation, okay, as I told you some time back. So if we take normal light, it has a two component. One is electric and magnetic. In the case of normal light, both magnetic and electric component are travel perpendicular to each other, 90 degree. Okay, but when light travel at a particular since it has two different components, electric and magnetic, light travels in a particular direction, that too with a particular speed. Okay, so light travels with a particular speed, then people start asking question, what exactly the speed of light, at what speed light is travels. Okay, so they have calculated the value is just 2.988 into 10 to the power 8 meters per second. Now it is being round into 3 into 10 to the power 3 multiplied by 10 to the power 8 meter per second. This is the normal speed of light. That is the speed at which light travels in a medium. So there are some relationship of light you should remember always when you go for spectroscopic analysis of any biomolecule. Whenever you're talking about light, you should remember first component, the speed of light. The second component is the wavelength of light that is being used, and the third one is called frequency. The frequency of light also very very important. So as I told you, each wavelength of light, let us take 300 nanometer wavelength of light, it possesses some energy. So the wavelength of energy is depending on the frequency of the light. Okay, there is a very strong relationship between wavelength, frequency, and also speed of light. The very popular formula is that c equal to mu lambda. So c represents the speed of light, lambda represents of course the wavelength, mu represents of course the frequency. If the frequency is very high, that light should have very high energy. The frequency of light is very very low, that means the light possesses very low energy. In terms of wavelength, higher the wavelength, lower the energy, lower the wavelength, higher the energy. The reason is that if the wavelength is very short, the frequency will be very very high. That's why if we take UV for example, 300 nanometer for example, or 280 or 300 whatever it is, let us take 280 for example. Okay, let us not confuse. Okay, let us take 280 nanometer wavelength of light. The wavelength is very short, therefore the frequency is very high, therefore the light is highly energetic. On the other hand, if you take wavelength of light in the visible region, that is called 700 nanometer wavelength of light. Okay, 700 nanometer wavelength of light, the wavelength is very high, so frequency is very very low, therefore energetically it is also very low. So 700 nanometer wavelength of light possesses very lower energy compared to 280 nanometer wavelength of light, which is having lower wavelength, higher the frequency. I hope you understand now the relationship between speed of light, wavelength of light, frequency of light, and also energy. Commonly we used to say energy lambda, wavelength of light, is indirectly proportional to energy. So higher the wavelength nanometer, lower the energy, lower the wavelength, higher the energy. So lambda is indirectly proportional to the energy. Okay, I hope you understand this basic concept without doubt. So there are some adduce or proof given by Max Planck as well as Albert Einstein for the particle nature of light. So they proposed what is known as a quantum theory of radiation, electromagnetic radiation. So what they have identified is they have identified the energy is transferred in discrete packets called quanta or photons, and they have also arrived at the equation which can relate frequency of radiation and the quantum also. So Q is equal to h mu. This is one of the very popular equation. Okay, so these two school of thoughts, one support the wave nature of light, another group supports the particle nature of light. Now we know for sure light has a dual property. Yeah, coming to the electromagnetic spectrum. Okay, excuse me. So assume sun emits light of different wavelength, which includes cosmic and gamma rays on left on your left up to radio waves. Radio waves are higher wavelength, cosmic and gamma rays are lower wavelength. So this whole spectrum is called electromagnetic spectrum. Okay, within the electromagnetic, if you look at the electromagnetic spectrum, within the electromagnetic spectrum, there is a narrow region of the electromagnetic spectrum which is considered to be very very important for biologists. Okay, that narrow region of the electromagnetic spectrum is called visible region of the electromagnetic spectrum. Okay, so the narrow region of the electromagnetic spectrum is called as the visible region of the electromagnetic spectrum, which merits attention by the biologist in general. Why, as a botanist or zoology, we are more concerned towards only the visible region of the electromagnetic spectrum compared to cosmic rays and gamma rays or radio waves? We neither worried about cosmic rays or nor the radio waves. So we are more concerned about only the visible region of the electromagnetic spectrum. The question is why? Everybody is asking, yeah. So why this happens? So this visible region of the electromagnetic spectrum is very very important with reference to biologists, because this visible region of the electromagnetic spectrum actually regulates plant growth and development. Okay, so this visible region of the electromagnetic spectrum regulates plant growth and development, which also influences the behavioral pattern of the some of the animal system also. That's why I use the term for biologists. This narrow region of the electromagnetic spectrum is very, very important. Okay, right. What exactly the nature of the narrow region of the electromagnetic spectrum? I hope this illustration will give you a better picture on the visible region of the electromagnetic spectrum. It starts with violet on your left and ends with the red. It is called VIBGR, okay, which includes violet, indigo, blue, yellow, orange, and red. If you look at very carefully this illustration, violet region contains very, very lower wavelength, okay, starting with 380 nanometer, and the red region, the far red regions, ends with 700 nanometer. So generally, while we are taking class, we used to say the visible region of the electromagnetic spectrum is in between 350 to 700 nanometer wavelength of light. If you look at very closely again, if you look at the violet region again, the wavelength corresponds to 400 nanometer, 380, 400. If you look at the energy in terms of represented in terms of kilojoule per Einstein, it is very, very, very high. That is 300 kilojoule per Einstein. On the other hand, if you look at the red wavelength of light, let me use the pointer so that yeah. So the violet, let, one second, I'll use the pointer. Yeah. So the violet has a wavelength of 380. Look at the energy; it is 300 kg kilojoule per Einstein. On the other hand, if you look at the red region of the electromagnetic spectrum, since wavelength is very large, look at the energy only 170 kilojoule per Einstein. So which confirms, so higher the wavelength of light, lower the energy; lower the wavelength of light, higher the energy. Okay. So just for our understanding, 1 Einstein is equal to 6 into 10 to the power 23 photons. Okay, right. So visible region of the electromagnetic spectrum is very, very important for biologists, which spans between 300 nanometer to 700 nanometer. Okay, right. Coming to the experimentation instrumentation component of UV visible spectroscopy. Okay, right. So as I told you, each biomolecule is capable of absorbing light of definite wavelength. Why a particular biomolecule is capable of light of only specific wavelength of light? Okay. So this phenomenon is relying on the structure of the biomolecule. So it is the structure of the biomolecule which dictates what wavelength of light to be absorbed by the particular biomolecule. Okay. So there is a very strong relationship between specific wavelength of light that is being absorbed by a specific biomolecule. Okay. So this is the instrumentation part of it. Okay, it's a simplified diagram of your UV visible absorption spectroscopy experiment. So the whole concept of UV visible spectroscopy rely on the absorption phenomena. We are not going to talk about light that is going to be transmitted, and also we are going to measure only the absorbance. Okay, we generally what we call it as optical density. So there are some concerns, okay, already developed in science, whether to use the term absorbance or the optical density. So there are some conditions, some circumstances where you have to use only the term optical density, where you cannot use the word absorbance. For example, you are trying to measure the, you are trying to construct the growth curve of the bacteria, okay, at different time points. Anyway, you are going to take the absorbance at only specific, by choosing the specific nanometer wavelength of light. In that case, you should not use the term absorbance. You have to use the word only the optical density, because what you are going to measure at a given different time points is only the density of the sample. Okay, that is the, which indicates the bacterial growth. So when you are trying to construct a bacterial growth curve, okay, by using UV visible spectroscopy, you should not use the term absorbance. You must use the term only optical density. Suppose if you have purified any biomolecule of interest, if you want to measure the absorbance of the particular biomolecule, in that case you can use the word absorbance. So you have to be very, very careful. Depending on the nature of your sample, you can use the word either absorbance or optical density. Okay, right. So this simplified diagram actually represents the basic hardware component of the UV visible absorbance spectroscopy. So there will be a light source which is followed by a monochromator, which is nothing but a prism. In between you have a pair of slit. The pair of slit will help you to select specific wavelength of light. Okay, and once the light passing through the second slit, it will impinges on the sample holder. This is called cuvette sample or cuvette holder. The light which is incident on your sample is considered represented as I naught. Okay, I naught. The light that is transmitted out of the sample or out of the cuvette is considered to be represented to be I. Okay, and finally, whatever light that is transmitted out of the sample can be measured by using the component of the UV visible spectrophotometer by name detector. So detector is basically working on the principle of photoelectric effect, but want of time I am not going to the details of photoelectric effect. Let me make it very simple. Whatever signal that is coming out of the sample, that is transmitted light, that signal is getting amplified within the detector. The basic mechanism behind the amplification process is called photoelectric effect. Okay. So what exactly we are measuring is finally the log I naught by I, which is considered as an absorbance. So the whole concept of UV visible spectroscopy depends on a Beer-Lambert's law, which actually calculate the log I naught by I. So the log I naught by I is considered as an absorbance. Okay, right. Depending on the the value of I naught by I, you can easily quantify the amount of sample present in your uh, amount of biomolecule present in your sample of interest. Suppose if I extracted protein, I can easily quantify how much protein is there in a given sample. Accordingly, I can calculate how much protein will be there per 1 gram of leaf in the case of leaf sample. In the case of any tissue sample, you can easily uh, calculate how much, if you take 1 gram of tissue sample, how much protein it contains. That can be easily quantified. Okay, everything depends on the Beer-Lambert's law, log I naught by I. That is what we are exactly actually calculated. Okay, right. There are three different types of UV visible spectrophotometers. Okay, the first category is called single beamed spectrophotometer, one of the oldest type of spectrophotometer. It is called single beamed spectrophotometer. As you can see here, the single beam spectrophotometer contain there will be only one light source. Oh, sorry. So there will be only one light source. Of course, there will be monochromator. This is nothing but a prism, which is separated by a pair of slit, and there is a cuvette holder and detector. Okay. In the case of, because of the advancement in science, then scientists have developed the split beamed spectrophotometer. The split beamed spectrophotometer is differentiated from the single beam in such a way that there will be one light source that will be passing through the single monochromator, and after passing through the monochromator, the light will be split into two components, so which will target two different cuvettes. One cuvette is corresponding to blank measurement, the another cuvette is corresponding to your sample of interest. This is called split beamed spectrophotometer. Okay, now whatever we are using in our laboratory, the advanced one is called dual beamed spectrophotometer. In that case, what happens, there will be two different light sources, or two separate light sources generated individual light beams. Both will target separately. One will target the sample cuvette, the another one will target the blank measurement. Of course, both the measurements can be reached only the detector for the quantification purpose. This is called dual beam spectrophotometer. So what happens in the case with reference to the price, in the case of price, the single beam spectrophotometer is very cheap. Split beam is relatively costlier than single beam. So now whatever we are using is called dual beam spectrophotometer. It is relatively very costly. Even the basic model, it comes around 4.5 to 5 lakhs. And talking about the best quality of spectrophotometer. Okay, right. So now this will give you the better picture now. Again, for visible light there will be separate light source. For UV light there will be separate light source. Altogether there will be a grating concept. Now we are using the concept called grating. Okay, grating will give you better light quality compared to conventional spectrophotometer. Okay, so there will be two different sample keyword, one for reference, that is for control, the other one is for sample keyword. Both can be selectively reach the detector depending on the absorbance of the sample. So finally everything is being computed now. So we need not do anything, just we have to keep the sample in a best quality within the sample keyword and then the reference keyword. Okay, rest of the thing will be taken care of by the computer itself. Okay, so in the case of conventional spectrophotometer, the general order of the play orientation of the optical component is light source followed by slit, then there will be a monochromatic prism, then there will be a sample, ultimately then you have the detector. From the sample, light passes through the sample and the whatever light that is being transmitted will reach the detector. This is the conventional optics of the conventional spectrophotometer. In the case of diode array spectrophotometer, one of the considered to be the advanced concept, from the light source sample will reach impinges on the sample first. So whatever light that is being transmitted, that is being dispersed by a monopolychromator, that is again the prism, by using the diode array spectrophotometer. So diode array spectrophotometer using the concept called grating. That's why it will give you the better picture of the light that is being transmitted out of the sample. So compared to conventional spectrophotometer, if you want to quantify exactly with minimum error the quantification of the biomolecule sample, biomolecule in your sample, you have to go for diode array spectrophotometer. So again, since the optics is very powerful compared to conventional spectrophotometer, the diode array spectrophotometer, dual beam spectrophotometer are very very costly compared to conventional spectrophotometer. Okay. So in the olden days we used prism. Now the prism has been replaced almost completely by holographic grating concept. The advantages is the light falling on the grating is reflected at different angles depending on the wavelength of light that can be captured by using the detector. So the sensitivity of holographic grating mechanism is very high, powerful compared to prisms. So as I told you, what exactly the physical basis of absorbance? So when a light of specific wavelength is impinges on the sample or biomolecule, the biomolecule absorbs light of definite energy, it will move to a higher energy. Okay, this is what happening in the conventional your UV visible spectrophotometer, which resulted in by the absorbance of the particular energy of light by the biomolecule of interest. Okay, if you look at the application aspects of the UV visible photophotometer, it has very wide application. For example, look at the panel A. The panel A represents, okay, sorry. So the panel A represents the absorption spectrum of adenosine monophosphate A. We call it as AMP. The lambda max is 260 nanometer. So there will be a sharp peak at the 260 nanometer. This is called lambda max. So the 260 nanometer is specific absorbance for AMP, adenosine monophosphate, which is being dissolved in a buffer with a pH of 5. So sometimes pH also influences the process of absorbance. You have to use appropriate buffer for the extraction of your biomolecule of interest. Your biomolecule has to be stable in that particular buffer with specific pH. Okay, so pH highly influences the structure of the biomolecule. So you have to be very very careful. Okay, so the lambda max for AMP is 260 nanometer, which means what? By using UV visible spectrophotometer, you can easily quantify the amount of AMP present in your sample of interest. Okay, look at the second one, B. It shows the absorbance spectrum. So whatever the product you are getting out of the UV visible spectrophotometer is called absorbance spectrum, which is nothing but a simple graph. Okay, which is being considered again using lambda wavelength against the absorbance. Okay, so the panel B shows the absorbance pattern of our phenylalanine solid line and zero point 0.7 millimolar histidine, Both are good amino acids. Phenylalanine is aromatic amino acid and zero point 0.7 millimolar. Sorry, histidine is a contains heterocyclic ring. Yeah. So the lambda max for this is 257 nanometer. So by using the simple UV visible absorption spectrophotometer, you can easily analyze the presence of phenylalanine and histidine. If phenylalanine and histidine is there, how much phenylalanine is there, how much histidine is there, that can also be done. So both it can be used as a qualitative analysis of phenylalanine and also for quantitative analysis of phenylalanine. And look at panel C, which represents the 70 microgram per ml of cytochrome C. Look at the lambda max, it is the 410. Okay, 410 is the lambda max for cytochrome C. So by using the standard also you can calculate, otherwise you can easily, by looking at the lambda max, the peak at the 410 nanometer, you can easily confirm the sample contains cytochrome C. Look at panel D, which represents the absorption spectrum of 65 microgram per ml of lysozyme. Lysozyme is a enzyme. Yeah. So look at the peak, it's almost a 230 nanometer. Oh, sorry, almost 230 nanometer. You can ask me now, okay, why the specific biomolecules are capable of absorbing light of only definite wavelength. Let us take example in this case. Let us take tryptophan. Tryptophan, the lambda max is 280 nanometer. In the case of bovine serum albumin, it is 280 nanometer. Why these biomolecules, both tryptophan and BVSA, can absorb light of only 280 nanometer wavelength of light? So tryptophan is an aromatic amino acid. The aromatic ring, any biomolecule with aromatic ring will absorb light only in the UV region. Okay, 280 nanometers is confined to ultraviolet radiation. Okay. On the other hand, you have a good amino acid, another good amino acid by name tyrosine. Tyrosine is also an aromatic amino acid, but it is not absorbing light at 280. The lambda max for tyrosine is only 274. Even minor variation in the structure of biomolecule results in absorption of different wavelength of light. Remember, tryptophan, tyrosine, and also phenylalanine all are considered to be aromatic amino acid, but all the three have different lambda max. They do not have same lambda max. The reason is that the structure of tryptophan is different, the structure of tyrosine is different, the structure of phenylalanine is different. But altogether, all are considered to be aromatic amino acid. They have a benzene ring in their structure. So the structure influences the absorption of specific wavelength of light. Okay. Of course, if you want to quantify DNA, you have to take absorbance at 260 as well as 280 nanometer. RNA again is a nucleic acid; it will absorb light at the 260 nanometer. Adenosine is one of the component of nucleic acid. Okay, that also absorbs light in the 260 nanometer wavelength of light. So since, as I already told you, the interaction of light with the matter is a quantum phenomena. That means it depends on the structure of the biomolecule. Okay, so each biomolecule is capable of absorbing light of definite wavelength. So what I have been taught even myself during my undergraduate or PG days, all aromatic amino acids will absorb light at 280 nanometer wavelength, but it is not so. Only tryptophan can absorb light at 280 nanometer, whereas tyrosine and phenylalanine, their light absorbs at 274 and 257 nanometer wavelength of light. But altogether, all these region are coming under ultraviolet UV region of the electromagnetic spectrum. Another important application of UV visible spectroscopy is that, assume you have a tyrosine molecule. Tyrosine molecule. If you add or if you remove one proton from the tyrosine molecule, it will be reflected in the absorption spectrum of the tyrosine molecule. Okay, you are removing just one proton from the tyrosine, deprotonated tyrosine. One is non-protonated tyrosine, another one is deprotonated tyrosine. That means you have removed only one proton from the tyrosine molecule. So removal of even one proton from the tyrosine molecule results in the different absorption pattern or absorption spectrum of the tyrosine. During this process, what happened? there is a concept called isobestic point. The literal definition of this isobestic point is that the wavelength where the absorption of both protonated and deprotonated forms of the chromophore are identical. Okay, so what is important here is, as I told you, each biomolecule is capable of light of definite wavelength. Why each biomolecule is capable of light of uh, why each biomolecule is capable of absorbing light of definite wavelength means each biomolecule consists of what is known as a chromophore. Okay, chromophore means it's a structural one of the domain of the structure, or one of the component of the structure of biomolecule which is capable of absorbing light. This region is called chromophore. Let us take protein for example. Protein contains a chromophore. You know, proteins are made up of amino acid residues. Amino acid residues, each two amino acids join together by a peptide bond. Okay, so once two different amino acids join together by a peptide bond, then it cannot be considered as an amino acid, it should be considered as an amino acid residue. Okay, so the peptide bond exists between amino acid residue of the protein is a chromophore for that particular protein. So peptide bond is capable of absorbing light of definite wavelength. Generally, peptide bond can absorb light in the 230 nanometer wavelength of light. 230 nanometer wavelength of light. So similarly, tyrosine has a chromophore. Your phenylalanine contains a chromophore. Your adenosine contains a chromophore. Your DNA, they do have a chromophore. Your RNA, they do have a chromophore. Because of the presence of chromophore only, these molecules are capable of light of definite wavelength. Is that clear for all of you? Right. So, if you look
Yes,
at
sir.
the
Yes, sir.
protonated absorption spectrum of tyrosine and deprotonated tyrosine, what I would like to emphasize here is, even you can analyze the presence of tyrosine whether it is deprotonated form or in the normal form. That can be easily differentiated by using the simple UV visible absorption spectroscopy. Okay, that can be studied by using this. Assume you have a biomolecule A. By some chemical procedure, you are trying to remove the proton from a particular biomolecule. Whether the proton is removed from this biomolecule or not, that can be tested by using simple UV visible absorbance spectroscopy. That is what I wanted to emphasize here. The difference between protonated and deprotonated form of a biomolecule can be studied by using UV visible spectrophotometer. Similarly, the presence of hemoglobin, a protein, in a particular buffer or particular sample can be studied by using UV visible spectroscopy. Even the influence of different solvents in the structure of the hemoglobin or any protein can be studied by using UV visible spectroscopy. The concept is called solvent perturbation concept. Okay, right. If you want to quantify nucleic acid, okay, you can easily studied, or you can easily quantify the amount of nucleic acid present in the sample of interest by using simple UV visible absorption spectroscopy. You have to take absorbance in both 280 and 260 and 280 nanometer. Okay. Not only that, the structural variation between the nucleic acid DNA can be studied. Whether DNA is undergoing degradation or not, that can be studied. Or what generally we call it as renaturation or denaturation of nucleic acids. Okay, denaturation resulted in the separation of two strands of the DNA. Okay, renaturation means annealing of both the strands of the DNA based on the complementarity. So renaturation and denaturation of nucleic acid can be studied by using simple UV visible absorption spectroscopy. So whatever I showed in this picture is called hypochromicity of nucleic acid. That means it results in decrease in the absorbance. During denaturation, what happens? If the DNA is intact, the level of absorbance by UV light is very, very low. When it undergo denaturation process, when the strands are getting separated, more adenine residues or bases are exposed to UV light, which increases in the absorbance. During renaturation, what happens? Because of the complementarity, the renaturation taking place, not much bases are exposed to UV light, so absorbance is keep on getting reduced. So the hyperchromicity or hypochromicity, simply speaking, denaturation or renaturation kinetics of nucleic acids can be studied by using simple procedure UV visible absorption spectroscopy. So as I have given here, the solid line indicates the concentration of free nucleic acid, the solid line, the single stranded DNA of the dotted line represents the concentration of single stranded DNA. The dashed line represents the double stranded DNA, where the absorbance is very, very low compared to the free nucleotides. Free nucleotides will have more absorbance compared to double stranded intact DNA molecule. Similarly, as I told you, even you can study the presence of protonated or deprotonated form of any biomolecule. In this case, NADH, within the sample, you can easily differentiate the presence of NADH and with that of the NAD plus. Oxidized form of NADH and reduced form of NAD plus can be studied by using the UV visible absorption spectroscopy. So the UV visible spectroscopy application can also be extended to study the enzyme kinetics. What exactly the rate at which enzyme bind to its substrate ultimately to produce a product, that can be studied by using simple UV visible spectroscopy. So whatever I have projected here is the kinetics of an enzyme by name alkaline phosphatase. Okay, so enzyme kinetics, that is the how much, what exactly the rate of enzyme, how fast the enzyme converts substrate into product, that can be studied by using simple UV visible absorption spectroscopy. The only thing is you have to use a specific wavelength of light that you should know before you start your experiment, for which literature survey will be of very great use. Finally, in a given sample, you can easily quantify how much protein is there. As I told you, know, I have been working with the tomato plant for almost 12 years. Okay, so we used to quantify even for our MSc project. Student MSc practicals, we used to quantify the amount of protein present in your sample of interest, especially leaf samples. Before you wanted to quantify the protein content of the tomato leaf, we need to construct what is known as a standard graph by using animal protein as a standard, bovine serum albumin. The procedure is very popular procedure, that is called Bradford method or Bradford assay. One of the highly cited paper in science is Bradford assay. Those who are working in protein, they must quote this paper, Bradford 1976 paper. Okay, so the construction of protein standard graph, UV visible spectroscopy is of great use, without which it cannot be possible. So 595 nanometer, you have to extract a protein, or you have to prepare a BAC at different concentration. You have to add equal volume of Bradford reagent, that is called CBB G250. You have to take absorbance at 595 nanometer. Okay, if you want to quantify a protease, there is an enzyme, okay, by name protease. If you want to quantify the amount of protease present in the sample, in that case you have to go for, you have to take absorbance at 650 nanometer wavelength of light. There is one more procedure for the quantification of protein, that is called Lowry's procedure. You will be adding a solution by name, you adding a reagent by name folin phenol reagent, that will also resulted in blue color formation. In that case, you have to go for absorbance at 650 nanometer. Bradford 595, folin phenol 650 nanometer wavelength of light. Okay. One of the finest application of the UV visible spectrophotometer. You can study even the structural denaturation kinetics are of the protein. The protein structures can be studied. Secondary structure of the protein can be studied by using the UV visible spectrophotometer. Okay, protein will absorb light with a different magnitude if it forms a native structure. If the native structure of the protein is disturbed, when the protein getting unfolded, then it will show a different absorbance kinetics. So the whether the protein is exists in the native form or in the unfolded form or denatured or in a refolded form, everything can be studied by using UV visible absorption spectrophotometer. So the application of the study of determination of structure is not only confined to only proteins. Any biopolymers, the change in structure can be studied by using UV visible spectroscopy. Okay, this I have already explained. No, this is just a thermal denaturation of DNA isolated from Pseudomonas aeruginosa. So UV visible spectroscopy is considered to be very one of the simple technique, but it has a very wide application aspect. So it has a lot of limitations also. Okay, that limitation I will try to give you the limitations of UV visible spectroscopy when I trying to talk to you about the fluorescence spectroscopy. Okay, why we are using fluorescence spectroscopy, why not UV visible spectroscopy? Okay, so if you take any biophysical technique, each one has its own advantages and also limitations. I won't say demerits. Okay, I will say only limitations. So with this I will finish the first part of my lecture, that is UV visible spectrophotometer. Okay,